Okay, we left in part 1 of DNA talking about the structure of DNA. And just a quick summary, DNA is a twisted ladder or a double helix. The sides or rails of our ladder consist of alternating sugars and phosphates. And there's our 5-carbon sugar here. Let me get my highlighter out. Okay, alternating sugars and phosphates. Attached to the sugars are what we call nitrogen bases. There are 4 nitrogen bases: adenine, thymine, cytosine, and guanine. Adenine and thymine are always attached to one another. Cytosine and guanine are always bonded to one another. So that's where we left off, talking about the structure of DNA. Back when we talked about mitosis and meiosis, we discussed how before a cell can divide, we have to be able to duplicate the DNA. We know that happens during interphase, during the S stage of interphase. And remember, the cell spends the majority of its time in interphase. And during that S stage, S stands for synthesis, DNA is replicated or copied. So big picture, we're using DNA to make more DNA. So this is number 8 on your notes organizer, which you should be filling in as we go through these notes. So big picture, replication is DNA being made into more DNA. So before a cell can divide, like I just said, a cell needs to be able to duplicate its DNA through a process called replication. By doing this, by copying the DNA, we ensure that our new cells each have a complete set of DNA. If we divide a cell without copying DNA, then we just end up with cells that have half the amount of DNA, which we of course don't want to happen. We want to make sure that each cell ends up with a full set, so we duplicate the DNA before we divide the cell. And of course, this takes place in the S stage of interphase. So in order for each new cell to obtain an identical copy of DNA, we have to sort of use our original strand of DNA as like a blueprint or a template. So the DNA molecule that's being replicated produces 2 identical new complementary strands following the rules of base pairing. So A is going to pair with T, and then G's are going to pair with C's. So the first thing we have to do, as you can see in this picture here, is we have to sort of unwind and unzip our DNA, and then we have to put the pieces of the puzzle back together so that the original strand serves as a template for the new strand. So each original strand of DNA serves as a template for the new strand. You can see in the picture here that the original strand is in blue, and it's serving as a template for our new strand, which is going to get put together piece by piece in our pink strand here. So that we end up with 2 identical copies of our original parent molecule of DNA. So here's a simpler picture showing that. This is called the semi-conservative model. Watson and Crick's model that they built made it very clear immediately how DNA was replicated. That this twisted double helix ladder unwinds, unzips, and then sort of using that unzipped portion as a blueprint gets put back together so that you have 2 identical molecules of DNA. This is called the semi-conservative model. Semi, because only part of the DNA molecule is conserved and used as a template or blueprint. So Watson and Crick showed the 2 strands of the parental molecule separate, and each functions as a template for synthesis of a new complementary strand. This is all number 8 on your notes organizer. So there's the parental DNA. It's being pulled apart, unwound and separated, unzipped, and then it serves as a blueprint for our new DNA. So we end up with 2 identical molecules of DNA. So just in summary, each strand of DNA, double helix, has all of the information needed to reconstruct the other half by the mechanisms of base pairing, and we'll talk about that in just a minute. There is a difference in how this happens in prokaryotes and eukaryotes. In prokaryotes, DNA replication begins at one single point and then continues in both directions. That's prokaryotes. Remember, organisms that don't have a nucleus. There's only one type of prokaryotic organism, and what is that? Bacteria. So in bacteria, DNA replication starts at one point and begins and continues in both directions. In eukaryotic organisms, so this is all other organisms, including humans, DNA replication sort of takes place all over at once. So it's happening in hundreds of places all at once and proceeds in both directions until all those little pieces get put together until the chromosome is completely copied. Okay, so where DNA sort of gets unwound, unzipped, and then where replication begins or occurs, that is called a replication fork. So here's a replication fork. where the split takes part and the pieces sort of get put back together in the puzzle. So this is still under number 8 in your notes organizer. So here's our original strand that at one point was together. Now it's been unwound and unzipped. And you can see piece by piece, we're going to have the molecule put back together, and that's happening in this direction too, until eventually we have 2 identical copies of our original parent strand. So the first part of DNA replication is unwinding the DNA, so now it just looks like a normal ladder, and then unzipping it right down the middle. Think about a zipper. You have the 2 parts that sort of get split right down the middle. That is done with the use of an enzyme called DNA helicase. I remember that one because it has the helix as the first part of the word, and it's unwinding the helix, so it's not a helix anymore. So it's responsible for unwinding and unzipping the double helix of DNA. And we have these little binding proteins that sort of hold the DNA open so that it can be replicated. But the one you need to know is DNA helicase, which is responsible for unzipping. Now, DNA primase adds a short segment of RNA primer, and that's really what begins the putting together process. However, the big enzyme here is going to be DNA polymerase. This is really what's responsible for putting together the new molecule of DNA. So we had DNA helicase that was responsible for sort of undoing everything, and now we have DNA polymerase, which is responsible for putting everything back together. Now remember, in eukaryotic organisms, this is happening in many places all at once. So we end up with these sort of little fragment pieces of replicated DNA. So you can see we've got a fragment here, we've got a fragment here, because here's DNA polymerase at all these different locations replicating the DNA. So we have to have something that puts all those little pieces together, and that's going to be the enzyme DNA ligase. So DNA ligase sort of is our last step in putting everything back together. Okay, so really quick, let's just summarize the process of DNA replication. First thing we need to do is we need to unwind and unzip the DNA. That happens with DNA helicase. The next thing we need to do is we need to put the DNA back together, and that happens with DNA polymerase. That is our really big enzyme that's responsible for putting everything back together. This is happening all at once in multiple places, so we create these little fragments called Okazaki fragments, named after the scientists who discovered them. And then eventually we need to put those fragments together to make one new replicated strand of DNA, and that's going to happen with the enzyme DNA ligase. Okay, so if this was a strand of DNA on our original DNA molecule that's going to serve as a template for the new DNA strand, what do you think the sequence of our new DNA strand might be, knowing that we have our base pairing rules where adenine pairs with thymine and cytosine pairs with guanine? So here's our DNA sequence, A, T, C, C, G, A, G, C, T, T. What would pair with adenine? That would be thymine. Then we have thymine here, so our complementary base would be adenine. Then we have cytosine here, so our complementary base would be guanine. Now you keep going and do the rest on your notes organizer, and then check it against my answer. So if we had DNA helicase that unwound our DNA, and this is what sort of was exposed, what would our DNA polymerase add to that strand of DNA? Here's our complementary sequence. Did you get it right? Good job. Okay, so last I'm going to go back to that picture so you can sort of label all of the parts of DNA replication. A would be our leading strand. B would be DNA ligase. Oh, I'm sorry. B would be DNA polymerase. C would be the parental DNA. D would be DNA helicase. E would be the tiny little RNA primer that begins the whole process. F would be DNA polymerase, the one responsible for putting it all back together. G would be the Okazaki fragments. H would be DNA ligase, the enzyme responsible for putting together the fragments. And then I would be the lagging strand. Okay, that's it for DNA replication. Hope you're having a great day.